introduce you a little bit, Alex, for people who doesn't know you yet. So I would use share screen. Oh, sorry. Alex, can you stop share?
Oh, yes.
I would like to share my screen first.
Okay.
Because I need this screen to introduce you is much more easy. So in Thailand, good morning for everyone, and it's good afternoon for Alex in Australia. So he is 3 hours behind us. Today is a really good opportunity to have Professor Loukas, our long collaboration for a long time, but some of the students probably doesn't know well. So I take this opportunity to introduce you as a special lecturer today. So Professor Alex Loukas is a distinguished professor at the Australian Institute of Tropical Health and Medicine at James Cook University, Cairns campus. Professor Loukas has been a collaboration with us, Faculty of Medicine, Department of Parasitology, and some other Department of Biochemistry and Department of Parasitology for a long time. I think almost 20 years that he started to collaborate with us. And he contribute with, working this research with us. And also he contribute as a supervisor for the overseas supervisor for the Royal Golden Jubilee student from the Faculty of Medicine. I think at least 5 students was working with him in the past. And one of the students of him, I think first student of him is me. And I was doing PhD with him as well. And today we invite him to give a talk on immunotherapy because Professor Loukas has been working with, his interesting is about the if you read and his primarily interesting is about the secretome of the parasitic helminth, and he used this worm secretes protein as a therapeutic and also diagnosis too. And he also working on the development of the vaccine. So his work is directly to this topic. And I think he is one of the world leaders who working on the immunotherapy using helminth. So this is a very good opportunity for us, Konkan University, for have him as a lecturer today. So I will stop here, Alex. So you can.
Okay. Thank you for the kind words. Let me see if I can share my screen. Okay, can you all see the PowerPoint presentation in full display?
It's full slide, but not the big one, Alex.
Oh, okay. I'm going to just unplug my monitor. Hang on a second. Okay. Sorry about that. Can you hear me okay?
Yep.
Just let me switch cameras for 1 second. Okay, and okay, can you see the slides better now?
Not yet. You haven't shared your slide, I think. I just see your face.
There's nothing wrong with that though, right?
Nothing wrong. Look okay.
Okay, is that better?
Yeah, but the window is on Zoom, not the right window. Okay.
Yep.
Okay, this is perfect now.
Okay. Okay. It gives me great pleasure to talk to you all. I wish I was talking to you in person in Konkan, but that will have to wait until May, which is not too far away. So what I wanted to talk about today was this concept of using controlled human hookworm infection to advance a number of different projects. What I'm trying to capture in the image you can see in front of you here is the different projects we work on in my lab, primarily around nematodes and flukes. I'm sorry for you folks in Konkan that this is a schistosome and not a liver fluke, but I very much enjoy working on Opisthochus. But what I'll really focus on today is how we have used controlled or experimental human hookworm infection, where we intentionally infect healthy human volunteers with hookworms to advance two particular projects, our interest in vaccines and our interest in immunotherapeutics. And we have, as Ajahn Deroda talked about earlier, we have a whole program on using worm molecules to treat inflammatory disorders, but I'm not going to talk about that today. I'm going to focus more on our work with experimental human hookworm infections. And then at another time, I'm perfectly happy, or if we have time at the end of the talk, I'm perfectly happy to talk a little bit more about the molecular work. And so as this diagram shows, we're really using, or not only we, but many others now are in the position to be able to use controlled Helminth infections. I'm talking about hookworm, but people are now doing this for schistosomiasis as well. And then others are doing it for whipworm, for trichurus infections, where you intentionally infect people so that you can monitor in a controlled environment the efficacy of things like anti-Helminth vaccines or anti-Helminth diagnostics. I don't have it on the slide here because we don't work with anthelmintic drugs, but people can now rapidly test the efficacy of new drugs or drug combinations by using these controlled Helminth infection models, where instead of doing everything in animals, you can actually very quickly go into a human trial to assess efficacy using these models. And then, of course, one of our main interests is the use of hookworms in particular as anti-inflammatory therapies, because we know the worms are really good at suppressing inflammation, and so they hold potential for treating a whole range of diseases that are a result of a dysregulated immune system. So diseases such as autoimmune diseases, metabolic diseases, allergies, and so on. So it's important just to sort of put things in context when we're talking about Helminth infections, and this is particularly relevant for hookworm. I appreciate that you would never do a human challenge infection with Opisthochus because of that link with cholangiocarcinoma, even though it's a result of chronic infection. There are some Helminths where it's just not safe or wise to do experimental Helminth infections. But for hookworm, at least, it's a little different. And even though everybody appreciates the downside of these neglected tropical diseases, such as the soil-transmitted Helminths, we know they cause crippling morbidity, thousands of deaths. We unfortunately do not have vaccines, and there are drugs for treating a lot of these illnesses, a lot of these infections, but there's a continual concern about drug resistance developing for human soil-transmitted Helminths, as it has many years ago for similar Helminths of livestock, where we already know a lot about drug resistance. However, on the other side of this scenario is a whole lot of benefits that infected people can experience when they have some of these infections. And again, this is really focusing on hookworm, but we know there are studies that we in my group and others around the world have done that show that when you infect somebody with hookworm, you increase the diversity and richness of their microbiome, and that associates with good gut health as opposed to poor gut health, where you have a lower diversity of microbiome. From numerous clinical trials, there's definitely evidence that there is a potential therapeutic benefit for a range of immune dysfunction disorders, and I'll talk about some of those soon. And then more recently, we know that helminths, and again, hookworms in particular, have a possible benefit in metabolic disease, and I'm talking here about metabolic syndrome and ultimately becoming type 2 diabetes, and I'll talk about that a little later as well. And so just to put this in perspective, this is very much, this map here is very much an oversimplification, but what we do know is that if you look at inflammatory diseases, so this is inflammatory bowel disease, one of the forms of IBD called ulcerative colitis. It's primarily restricted in its distribution to westernized developed countries where there's been a lot of industrialization, and there's a great reduction in infectious diseases in these countries as a result of their urbanization. So even though this is IBD, this same distribution applies to many of the autoimmune and allergic diseases. If you look on the other side here, this is soil-transmitted helminth infections, and again, this could be many of the different helminths or parasites that are issues primarily in tropical countries. And so you can see here, obviously, the tropical regions of the world, and particularly those that are underdeveloped, have the highest incidences, in this case, of soil-transmitted helminths. But again, it applies for schistosomiasis and a bunch of other parasitic diseases. And what you'll notice, these are almost the complete opposite of each other in terms of their distribution. And what we know in this area is that these immune dysfunction disorders are certainly less common in developing countries or countries that haven't been fully urbanized yet. And what we also know is that worms, or you can think of them as the macrobiome, as opposed to the microbiome being the gut bacteria. Macrobiome is pivotal in educating our immune system. And so when children first experience helminths and other infectious agents, but particularly helminths, the immune system receives triggers to deal with the infection, but also to induce a form of tolerance so that you have a very well developed regulatory arm of the immune system, as opposed to the inflammatory or effector arm, which has been devised to attack infectious agents. And really, it's only very recently, in terms of evolutionary history, that we've removed these helminths from industrialized countries. It's really only 100 to 200 years, or even less in some places. And so what's happened is very rapidly our immune system has been deprived of essential signals that develop the regulatory arm of the immune system. And this has resulted in a massive expansion, again, particularly in developed countries, of autoimmune diseases and allergies and metabolic syndrome in particular. And so numerous people, including ourselves, have asked this question: can we reintroduce worms to treat or prevent some of these diseases of modern society, these inflammatory diseases? And one of the first people to ask that in terms of doing clinical trials was this guy here, Joel Weinstock, who's a gastroenterologist who lives in the US. And he worked with a company called Coronado Biosciences, who were trying to develop therapy with the pig whipworm, Trichuris suis, as a treatment for inflammatory bowel disease. This time they were focusing here on Crohn's disease, which is a different form of IBD to what I talked about earlier, ulcerative colitis, but still the two major forms of IBD, inflammatory bowel disease. And being a gastroenterologist, this was his area. And so they formed this company, and they manufactured, or they collected Trichuris suis eggs or ova from pigs, and they cleaned them up with CGMP processes, and they administered them to patients in a phase 1 clinical trial, which was published and showed very encouraging efficacy. But then when they did a larger phase 2 clinical trial in 250 patients with moderate to severe Crohn's disease, the study found that it did not meet its primary endpoint, and that was improving the response to the disease. So this was disappointing and a bit of a surprise to the field. And there were numerous thoughts about why it didn't work, but my perspective on this is that these guys focused on the wrong worm. They were using a worm that does not mature in people. This is a pig whipworm, and if you ingest the eggs, which is the infective stage of Trichuris, you can see here the eggs with the L1, the infective larva developing inside. When the egg hatches and the L1 comes out, these larvae only live for a few days or less than 1 week in humans because they're not adapted to survive in humans. And so that right from the beginning, that was a problem. And it might be a good business case because people have to take this medicine here with the eggs. People have to take it every week. So you sell a lot of your drug, but at the same time, these worms do not establish and grow to adulthood and develop a chronic infection in people. And what we know from you've probably heard in your classes about parasite immunology is that when these infections become chronic and long-term, that's when the real immunoregulation kicks in and the worms induce an immune response that does not get rid of them, but protects them and allows them to live for long periods of time without doing too much damage to the host. And so Trichuris suis does not really get a chance to do that in people because it does not fully mature. And so I have a colleague here in Australia called John Cruz. He's a gastroenterologist as well. And he had the idea quite some years ago now. This paper is from 2005. So it's almost 20 years old now. But he had this idea of why don't we do this with hookworm, Nicata americanus, because some studies that happened before we got involved had shown that it's reasonably safe and well tolerated in human volunteers. And it establishes a chronic infection in people and lives for many years, which is obviously very different to Trichuris suis, which is a zoonosis, and it's not a true human parasite. And so John, a number of years ago, conducted a small open label study. What we mean there is that's a clinical trial without a placebo group. It was just every patient who enrolled knew what they were receiving. There was no blinding or randomization. And all the patients were just given a low dose of hookworms. I can't remember the exact number of worms, but it might have been between 20 and 30 worms. And after those worms established in the gut, in general, there was a trend towards reduced Crohn's disease activity in the gut. What you can see here is this is the Crohn's disease activity index, and anything below the red line is considered by gastroenterologists to be in remission. And so you can see some of the patients had active disease at the beginning of the trial. Some of them were in remission, but at the end of the trial, of those who remained for the full year, those who remained in the trial were all in remission and did not have active disease. So this was really encouraging for us, but there are numerous reasons why inflammatory bowel disease is difficult to work with, and primarily you can't control the disease. People have flare-ups and they're very sick, and then they get better and they can remain better, and no one quite knows what really triggers these episodes of severe disease. But there's another disease that you can look at that has very similar pathology to inflammatory bowel disease, and that's called celiac disease. And I actually don't know how much of a problem it is in Thailand, but certainly in my country and in Europe and the US, celiac disease is a big problem. And it's an autoimmune disease. It's not an allergy. It's an autoimmune disease, but it's triggered by consumption of gluten in the diet. And gluten, if you're not familiar with what gluten is, it's a major protein found in wheat and barley and rye. And so typically, if people who have celiac disease eat any gluten, even just a small amount of pasta, like you can see on this plate, that's enough to make them very sick. And so in 2015, we did a clinical trial. Again, this was an open label trial, so no placebo group. And we took people with, this was about 12 people, I believe, and we gave all of them just 20 hookworms, just a small number of hookworms. We applied the larvae, the infective L3 stage larvae, to the skin, and those parasites entered the body, and established a chronic infection in the gut. And then over a number of weeks, this is weeks in the blue bar, we slowly introduce gluten back into the diet of these people. So people who have celiac disease are very good at managing their disease. They just avoid eating gluten and they eat foods that are free of gluten. And these people were on a gluten-free diet. And then 12 weeks after their worms had been applied, we slowly introduced gluten back into their diet. This much pasta, which is the equivalent of 10 milligrams of gluten each day. And then we stepped it up to 50 milligrams of gluten. And then at this point here, 24 weeks, we stepped it up to 350 milligrams per day. And then the trial was supposed to end here at 36 weeks. But at this time point, nobody had gotten sick. Whereas we know from previous studies that if people don't have worms and you give them this much gluten, they get very sick in general. And so we went back to our ethics committee and asked them if we could give these people even more gluten. And so during this period here, while we were talking to the ethics committee, the people went back on their gluten-free diet. And then for 2 weeks here, we gave them this much pasta every day. And again, none of them got sick. And so that was really interesting to us. What we effectively had done in this trial was to reduce or to improve the ability of people with celiac disease to eat gluten. And that was because they had worms on board. You don't need to worry too much, but this is just how we scored the pathology of these people. When you have celiac disease, the state of the active state of the disease is a grading of what the villi, how tall the villi, the microvilli in the gut are, and other scores as well, including the number of intraepithelial lymphocytes in a biopsy. And so biopsy tissue has to be removed from the gut by a gastroenterologist to diagnose the state of the disease. And that was great because it allowed us to do some immunology in the lab with human gut biopsy tissue instead of just blood. And you can see here before the trial, after the first low dose gluten exposure and then after the heavy gluten exposure, we did not see any change in the number of T cells in the gut, which was a good sign. And interestingly, when we look at different types of T cells, particularly these interferon gamma producing T cells, which we know are the main cell type that drives pathology in the gut in celiac disease, we saw over time that those T cells, those interferon gamma producing T cells came down beginning of the trial versus the end of the trial. And interestingly, in the opposite direction, we saw this different family of T cells. These are called regulatory T cells. They express this marker called FOXP3. These are the cells that actually prevent inflammation and stop these guys from functioning here. So this is like the holy grail in autoimmune diseases, is anything that can increase the expression of regulatory T cells to suppress inflammation. And we saw those numbers go up in the gut of people over time during the trial. That was really interesting to us. And just before I move on, I just want to show you a video. You recover the pieces of gut tissue from the small bowel to diagnose the celiac disease status. Worms. Here you see hookworm. Here she's attached. This is blood inside the gut. The male hookworm is a bit smaller and attached together. And we're looking down on the top of them in the gut. But before I press play on the video, what's remarkable about this image is that you have 2 1 centimetre long worms here feeding blood, but there is no inflammation around them. There's no redness, there's no blood leaking out, there's no ulcers. This is a very healthy gut. And what that told us is that even though these worms bury their heads right under the mucosa, deep into the tissue so that they can suck blood, what it tells us is that these worms have remarkable properties to be able to regenerate the mucosa and prevent tissue damage. And you can now just see here. But what's interesting is this little red mark here, this small ulcer, and my gastro colleague believes this is probably where the worms were attached a few hours. They let go and they move. And then if he said if he would come back, that he would not see this. It would have full estimate to show you how potent these worms are at suppressing inflammation. That's a very healthy looking gut, despite the fact that you've disease. So we use that information to get a grant to do a much larger clinical trial. And this one was a randomized placebo-controlled trial where we had groups of people who either received 20 infective larvae or 40 infective larvae or placebo. And the placebo was just, we put these larvae on the skin of people and they create an itching sensation when they penetrate through the skin. So here we just provided some chili sauce on the skin to create a mild itching sensation, which was as close as we could think of to the sensation. You don't need to worry too much about the details here. This is how we administered the worms. But what is important is the next slide, and that is that the results weren't as impressive in this placebo-controlled trial as they were in our, was that people who had worms felt better, even though they weren't sure if they had worms or placebo. They did feel better and they stayed in the trial for longer. One of the things that was a problem in this trial was that it was a multi-centre trial. We had trial sites in Australia and in New Zealand, and in the New Zealand site, we had to transfer worms from Australia to New Zealand, and even though it was done with a temperature-controlled environment, some of those develop eggs in the feces, and they did not develop eosinophilia, the type of white blood cells, eosinophil white blood cells that are a typical marker of most Helminth infections. So we think that some of the people in the New Zealand site, their treatment did not work. They did not become positive. And so if we separated the data, you can see if you look at how long these people lasted in the clinical trial, people who were hookworm positive tended to last longer. So that means that they did not drop out of the trial because they started to feel sick when they started eating gluten. And also, when you're doing, they fill out a diary every day about how much pain they are in their gastrointestinal tract. And again, the hookworm negative people in red here had higher gastrointestinal. So this trial wasn't as encouraging as we'd hoped, but there was certainly a suggestion that there is something going on there and that hookworms do have at least some therapeutic benefit in celiac disease. Of course, what we'd like to do in the future is do a larger number of worms. In this trial, we did not go above 40 worms. And since we've published this work, colleagues in the Netherlands have shown that the subjects in their clinical trials have been able to tolerate even 100 worms very well and don't describe any more symptoms than people who get low, and they certainly do not develop any signs of switch, which is a concern for some people with uncontrolled hookworm infection. So I want to switch now from, and you might think, well, celiac disease is an autoimmune disease. Why is he now talking about type 2 diabetes? Type 1 diabetes is an autoimmune disease, but I'm not talking about type 1 diabetes here. We're actually interested in type 2 diabetes, which is a major problem in Australia, in Europe, in America, and it is on the increase all throughout Asia. And that's primarily due to our lifestyle, but as well, it's also due to our decreased exposure to a whole lot of different infectious agents, including helminths. And in recent years, it's really been recognized that type 2 diabetes is a metabolic disease, but it also has an inflammatory component. We know that the immune system is altered in people who are obese and have type 2 diabetes. And particularly, you get a whole lot of inflammatory cells accumulating in the adipose tissues, the fat tissue, the liver, the pancreas, and even in the circulating blood, the vasculature of people who are diabetic. And what this does is it results in altered levels of inflammatory cytokines. And you can see here a picture of when there is high levels of glucose in the gastrointestinal tract, the adipocytes, and you get accumulation of these inflammatory cells like T cells, mast cells, macrophages. These M1 macrophages accumulate in the adipose tissue, causes beta cells in the pancreas and the pancreatic islets to produce inflammatory cytokines, again, like IL-1 beta and some of these chemokines. And what that results in is a pro-inflammatory environment in these tissues and also in the liver in people who have type 2 diabetes. And preliminary results, actually it's not preliminary now because this is from a review from quite a while ago now, but in a number of clinical trials, different anti-inflammatory drugs have shown potential to improve type 2 diabetes and insulin resistance, including some of the aminosalicylates and even antagonists of interleukin-1 beta, this cytokine that really drives a lot of the immunopathology. And so there's really a need for immunomodulatory strategies to treat the disease that both lower blood glucose levels and also potentially reduce the severity and prevalence of the various complications in type 2 diabetes. We know from work that predates my involvement in the field, there's been a lot of work over the Celiac syndrome, particularly rodent studies, and more recently some human studies. But work that we've done in my lab where we've used a mouse model of hookworm infection with a rat nematode called Nipostrongylus brasiliensis, which is very similar to the human hookworms. If you infect mice with these Nipostrongylus parasites and you put those mice on a high-fat diet, over time, those mice will become very obese and grossly enlarged, and after 6 months, they are fully diabetic. And if you look here, this is the blood glucose levels of mice that are on a high-fat diet but are uninfected, compared to down here, the red line is the blood glucose levels of mice that are on a normal diet but are infected with Nipostrongylus brasiliensis. We see the same thing, the black squares is the naive mice on a normal diet and the infected mice on a normal diet. This line up here at the top is uninfected mice that are on a high glycemic index or high fat diet. But if you take mice and feed them the same diabetes diet, but they're infected with Nipostrongylus, you can see you get much lower blood glucose levels. And that's whether you take this, whether you start to treat the mice with the parasite before they get the high-fat diet or after they have already gained weight from the high-fat diet. So this infection has both therapeutic and prophylactic properties. And interestingly, lots of studies of immune response that ones induce, which is generally referred to as a T helper type 2 immune response, seems to underpin all the change, because some have shown that eosinophil deficient mice lose this protective property, and you can also transfer some of the protection with various Th2 cells and Th2 cytokines. And so what we think happens is you've got a helminth. This is a hookworm, for example. We know that early innate immune cells to produce alarmins like IL-33, which act on innate lymphoid cells, and then these innate lymphoid cells secrete cytokines like interleukin 5, is a recruitment factor for eosinophils. Immune cells are found in the adipose tissue and these M2 macrophages, and they create more of an anti-inflammatory. What you see in obese have M1 macrophages, and you get this inflammatory state. Whereas in worms and doing early stage clinical trials, we thought it would be good to see if what we see in the mouse actually happens in the humans. No one has ever done a clinical trial in type 2 diabetes with worms. There's some psoriasis endemic areas where if you treat people who are naturally infected with anthelmintics, to actually done it in a careful clinical trial setting. And so my colleague here at JCU, Paul Jackerman, he ran a clinical trial, which was a randomized, double-blinded, placebo-controlled trial, where we had metabolic syndrome. So they had 2 of the 3 required clinical indicators of type 2 diabetes, and one of insulin resistance per group into the placebo group, a group that got 20 hookworms and a group that got 40 hookworms. And we apply the hookworms onto the skin here in a drop of water, because the L3 stage is as opposed to the adult worm. These were the inclusion criteria for the clinical trial. They were adults between 18 and 50 years old. They could not be pregnant, so that was an exclusion criterion. 20 centimetres for women and more than 102 centimetres for men. They could not have any of these diseases. They needed to have insulin resistance as assessed by this, this homeostatic mechanism that's been predetermined by endocrinologists. And they needed to have at least 2 features of metabolic syndrome, including elevated blood pressure, dyslipidemia, or abnormal liver function. And so the study looked like this. We applied once people had enrolled, we collected blood. We did a body scan to look at their body fat, and collected feces and had a physical and questionnaires. And then we gave them their hookworms on week 0 and also week 8. So people who got 20 worms got 10 worms on week 0 and another 10 worms at week 8, or the people who got 40 worms in total received 20 and then another 20. And then we followed them for 2 years, and all these different things happened at different points of time. And the primary outcomes of the trial was safety and tolerability. So did the people tolerate the infection, and was it determined to be safe? And the primary metabolic outcome was insulin resistance. So did the worms improve insulin sensitivity? And then there were a bunch of secondary outcomes, which we are looking at all of these now because the trial only ended a few weeks ago, but we do have this data, and I'll show you in a minute. So interesting, and we were delighted with the outcome. If we look at insulin resistance, this is the change in insulin resistance units. The people who had hookworms had significantly reduced insulin resistance compared to the placebo group. With hookworms, their insulin sensitivity improved as a result of the infection. Now, it was not statistically significant, but we certainly saw a trend to reduced weight. so weight loss in the people who had the hookworms, and a small change in weight, a gain in weight of the people in the placebo group. But this did not reach significance. So that's as much data as I can share with you at this point in time from that diabetes trial, but we are analysing all the other data, including immune response data now, and we'll be attempting to publish that work soon. But we were delighted with that, and it's certainly stimulated a program in the lab where we are now looking for the actual molecules that drive that Th2 response in people, and thinking about how those might be used as a therapy for people with diabetes in the future. Just for the last 15 minutes of the talk, I just want to now switch gears, and instead of talking about using worms to treat inflammatory conditions, I want to talk about trying to develop a hookworm vaccine to kill hookworms instead of using them for good. And that's because people in developing countries, and particularly in areas of sub-Saharan Africa, who get exposed to hookworms have very high worm burdens, they have very poor diets, and so they actually become anemic as a result of losing too much blood from having too many hookworms. So even though there are drugs available, we clearly need to have a vaccine, because you need to treat so frequently with those drugs that it just doesn't become possible. And I don't think there will ever be elimination of a helminth, at least in areas like Africa, by relying on drugs alone. So we are interested in developing a hookworm vaccine. And here you can actually see Nicata americanus larvae attempting to penetrate through parafilm. This is that wax film you would cover your tubes with in the lab. If this was actually skin, they would get through the skin very quickly. So just to remind people of the life cycle of the hookworm, because this is relevant for developing a vaccine, we believe that the ultimate hookworm vaccine will target multiple different developmental stages. And of course, this also probably applies to any Opisthochus vaccines that are developed. Any worm with different developmental stages has different ways of evading immune attack. And so the more points in the life cycle at which you can target, the better. And so for hookworms, we're very interested in targeting the larvae penetrating the skin and getting into the lungs, and the adult worms that live in the small bowel. And so keep in mind this L3 stage, which usually you find in the vegetation or in the soil in hookworm endemic areas, it penetrates through the skin, and that's the way we infect our volunteers. And so we thought before we try to find a vaccine based on proteins, a subunit vaccine to get rid of a human helminth, why don't we think about going back to the very beginning and just using irradiated parasites? It's been done with various helminths in animal models, but no one's ever done it in humans. And so we did that. And this is just showing you some of you guys probably already know this from your backgrounds in parasitology, but there are very few successful vaccines against helminth infections that are available on the market, and they're all for animals. There's none for humans. And of those that are on the market, the ones for nematodes like cattle, cattle filarial nematode, and there was a hookworm vaccine many years ago that was available for dog hookworm, Ankylostoma caninum. Those vaccines were actually irradiated infective larvae, and that was what the actual vaccine was. There are recombinant vaccines for a few helminth infections, but they're all cestodes, they're all tapeworms, and they have proven to be easier to kill immunologically than nematodes, particularly because a lot of these parasites here, the host becomes resistant, immunologically resistant to infection after just one round of exposure. And so these vaccines are just accelerating a natural process that would occur when these animals, sheep and other and cattle, once they mature, they become resistant, and these vaccines just accelerate that process. These various nematodes, however, are much harder to kill immunologically. And so if you ask the question, why are there so few vaccines for helminths? It's because there's many reasons, but these worms are very sophisticated. They have huge genomes. They're very complicated, different developmental stages that we talked about earlier. And any single component subunit vaccine, so a vaccine that relies on just one protein and an adjuvant, will only really work with these worms that are easy to kill, like these tapeworms. And what we really need is multivalent vaccines that target different processes and different developmental stages. Why aren't these irradiated vaccines being developed for humans? Well, no one's ever tested them until now, but also it's going to be very hard to scale these up. The way you obtain these larvae is by collecting feces from naturally infected hosts. And you would need a lot of parasites from a lot of experimentally infected human volunteers to be able to make such a vaccine for humans at large scale. So we decided that we would go back to basics. There are some hookworm vaccine antigens that are actually in clinical development and have been tested in phase 1 clinical trials called APR1 and GST1. These vaccines have only been tested for safety. They've never been tested for efficacy, so we don't know if they actually will work. My guess is each vaccine antigen alone will probably not provide sufficient efficacy. And now that there is a human challenge model, we think that this accelerates the ability to test these vaccines instead of just testing them in the field in endemic countries. We can now actually, in tightly controlled environments, we can test these in previously helminth naive individuals. And again, in terms of irradiated hookworm larvae, we know that there was one that worked in dogs and it was available commercially for a few years in the 1970s. It was taken off the market for various reasons, but one of those reasons was not poor efficacy. It was actually quite a good vaccine. So we did such a trial with colleagues in Brisbane at the Queensland Institute of Medical Research, Paul Chapman, who's an infectious diseases physician, and my colleague from many years now, James McCarthy, who's also an infectious diseases physician. And we took Nicata americanus larvae that we have in numerous volunteers here in my lab, and we irradiated them using ultraviolet light and conducted some in vitro penetration studies to see if we had attenuated those larvae enough so that they were still alive, but that they were compromised. And what you're looking at here is we had control larvae that were not irradiated, and then we had 2 different groups of larvae that received irradiation. We called them vaccine 1 and vaccine 2. And then we had another group of larvae that we used to challenge people. And so these were non-irradiated larvae. And if you look on day 1 and day 14, you can see that at day 1, both the irradiated and the non-irradiated larvae were all mostly motile. This is how much they moved. And then 2 weeks later, you can see the 2 groups of larvae that received attenuation by UV, they were much less motile than the 2 groups of larvae that were not irradiated. And this was great because we wanted to be able to slow these worms down, but not kill them so that they were completely immotile, because these worms needed to be able to survive inside the host, induce an immune response, but not develop to become adults. So our clinical trial looked like this. Ignore this top one. This was the dose finding study. This is what we did down here. So on day 1, people received either the placebo, which again was chili sauce on the arm, or they received UV attenuated hookworm larvae. And then they received a dose of albendazole, so the drug that kills hookworms, 4 weeks later, just to make sure that none of these parasites that had been attenuated had actually made it into the gut. We wanted to make sure that there were no adult worms in the gut. And then we gave people a second inoculation on day 42. And then we did the same thing, another dose of albendazole. So this here is the vaccination phase, 2 exposures of irradiated larvae, or the placebo group who just got chili sauce on the arm. And then part B down here is the challenge phase. So once people had been vaccinated or treated with placebo, we then gave them an inoculation of larvae that were not attenuated. These were healthy hookworm larvae, no UV radiation. And then we followed them. This was day 84. Then we followed them for a bit over 100 days. At the end of the study, we collected feces and blood all the way along here. B equals blood, and you can see how often we did that, and F is feces. And then at the end of the trial, everybody had albendazole for 3 days, and that was the end of the study. So the things that we monitored throughout the trial was, of course, one of the things we had to monitor was adverse events. And one of the adverse events of hookworm infection is the effect of skin penetration. And we measured that in 3 different ways: induration, erythema, and the duration of the dermal symptoms. And I'll show you photographs in a minute of what I'm talking about. But the first vaccination in both the placebo group and the vaccine group, you had no effect on the skin. The second vaccination, the vaccination group started to respond with skin reactions. Of course, the placebo group didn't, because they did not get hookworms. And then at the challenge with the non-irradiated helminths, you can see every participant responded on the skin, but the challenge group, the placebo group, mostly did not. Only one participant responded here, because this is the first time this group's been exposed to hookworms. We can see the same for erythema, but we see a response a little earlier. And then the duration of the symptoms. Interestingly, the more people were exposed to worms, the shorter the duration of the symptoms. And this is sort of indicating that over time, people develop tolerance to the exposure. This is what some of those dermal reactions look like. This is day 3 after someone's received 50 irradiated larvae. You can see some red marks. Each little red dot here is where one larva has probably penetrated the skin. And here there's been a collection of larvae. On day 44, so after the second inoculation of the same subject, so this is day 1 of subject R405, after their second inoculation, you can see the response is exaggerated, it's worse. And you get these small blisters appearing. This was not painful for the participants, but it was very itchy. So it was intensely itchy, and some of them had to put steroid cream on the wound, and you can see the white steroid cream here, because it was just so itchy. Excuse me. Over the duration of the period, we also detected eosinophils in people. And again, you probably all know that eosinophils are an indicator of Helminth infections. And again, the vaccinated people in black, the placebos in red. You can see that the 2 vaccinations, here's vaccination 1 and here's vaccination 2. The vaccinations tended not to induce much of an eosinophilia. It wasn't until the challenge phase with non-irradiated parasites. These are the parasites that actually get to the gut. It wasn't until worms started to really get to the gut that we see eosinophils in the circulation take off. And of course, you can see here the placebo group. This is their first exposure to worms, is here at the challenge stage, and their eosinophilia started to rise, but not as high as the people who had received the 2 doses of the vaccine. Now, the thing that everybody wants to know is, did the vaccine actually work? Did it cause reduced numbers of parasite eggs in the feces compared to the placebo group? When we looked at the eggs in the feces, we saw a trend, but we didn't see a significant decrease. Keep in mind, this was a very small study. We only had 6 people in the placebo group, 5 people in the placebo group, and only 4 of them actually provided samples, fecal samples, so that we could measure the hookworm DNA in the feces. This is based on an assay from James McCarthy's lab. That's a qPCR for assessing the intensity of hookworm infection compared to using microscopy-based egg counts. But you can see here in the vaccine group, we'd had a trend towards reduced DNA in the feces. But where things were more exciting was when we took those eggs from those vaccine group or the placebo group, and we hatched the larvae from those eggs. So when we collect eggs from these vaccinated individuals and the placebo individuals, we culture them and allow the eggs to hatch. And then we count the number of L3 that come out. And essentially what we saw here was a significant decrease in the number of L3 that we recovered from the eggs from vaccinated individuals compared to placebo individuals. Even though there was only a small number of people here, this was a significant outcome and we had bona fide clinical statisticians do that analysis for us. So even though this was a small study, it's the first example anywhere where there's been a clinical trial in humans with experimental Helminth challenge and an actual vaccination that assessed efficacy. And even though it was a very small trial, we believe this is an example of early stage efficacy. And of course, now we want to do a much larger trial to see if it holds true. This is just showing that the vaccinated participants made an antibody response over the duration of the trial. This is an ELISA in response to Necator americanus L3 antigen extract. We are now looking at individual antigens, but we don't have that data yet. Interestingly, the vaccinated participants also made a very strong cellular immune response to the L3 Necator extract. You can see here both Th1 cytokines along the top here, interferon gamma, TNF alpha, interleukin 2. We got a strong response, particularly for TNF and IL-2 in the vaccinated, but not the placebo group. And as expected, we got a strong Th2 response, IL-4, IL-5, and IL-10. IL-4 and IL-5, we expected. These cytokines have been associated with reduced Helminth infections in some settings, with hookworms in particular. IL-10, we didn't see a significant increase, but that's not unexpected because this is a regulatory cytokine as opposed to an effector T cell cytokine. So what do we think an ultimate hookworm vaccine looks like? As I've already alluded to, irradiated larvae are difficult to work with. I can't imagine how you would collect enough to scale up and make enough vaccine to treat millions of people. It was easy enough for us to do for a small number of people. It's tough to do on a larger scale. Also, it would be much harder to get regulatory approval and develop CGMP methods for this. We and colleagues are trying to do it at the moment, but it's just not an easy, feasible approach. A neglected tropical disease vaccine really needs to be cheap to make and easily manufactured, because the people who need to buy it are the people who can least afford it. And so what we're trying to do with colleagues at University of California, Irvine, is use protein microarrays to identify the antigens that are the major targets of the protective immune response. So we just sent serum from our clinical trial subjects to our colleagues in California who have made a Necator americanus proteome array with 300 secreted proteins on there, recombinant secreted proteins. And they are now going to probe those proteins, those protein arrays, to identify which are the major IgG targets of our vaccinated subjects. And we can then explore those recombinant proteins in different formulations in animal models. So where are we heading with this? We are wanting to do bigger and better powered studies to really see if the early indicative efficacy we saw holds true. We will do a higher dose of larvae, a higher number of UV attenuated larvae for our vaccination, because we went very carefully in our first trial. And we'll also challenge people with a higher number of larvae, so that if there are benefits there, we'll see it when we do a larger challenge. And as I've said, we want to identify the protective antigens using immunomics approaches and test them in animal models. And we're also very interested, and this is work we've been enjoying with you folks at ConKen as well, is this idea of oral vaccines for Helminth infections. And with colleagues at the University of Queensland, we have made some oral formulations of hookworm peptide-based vaccines and shown that they are indeed quite efficacious in mice. You can see here, this is the number of worms in mice that received an oral formulation of a hookworm vaccine, P3 lipopeptide. Sorry, this one here, P3 liposome, compared to a non-hookworm peptide from a bacterium. So this is showing great early efficacy. And we've just had another paper published with these colleagues from the University of Queensland showing again that other oral formulations work in animal models of hookworm. So the ultimate hookworm vaccine for humans is probably going to be based on peptides and proteins against multiple targets, and hopefully an oral formulation. And so I think I might just leave it there. I'll skip that last slide. Many people in my lab have contributed to this work. And of course, key collaborators who I talked about, especially the vaccine guys from Queensland Institute of Medical Research in Brisbane, and our colleagues at University of Queensland who do the oral formulation, and our clinical collaborators, the gastroenterologists in Brisbane, and our colleagues in California. I'm going to leave it there, and I'm happy to take any questions that you may have.
All right. Thank you very much, Alex. Very informative lecture, both on vaccine and immunotherapy. I think this would be generate a lot of questions. I would like to request questions from students first. Any questions from students?
If people don't want to ask questions, they can put them in the chat.
Yep. I think there would be some questions. Waiting for a minute. So while we're waiting for a question from students, I have some question, Alex. If you compare the immune response, I mean for treatment in between like low level, like in the laboratory you did in Western country, and in the normal infection state, are there difference, or in the normal infection would be already have protective for that kind of inflammatory disease or whatever? Have you investigated that?
I'm not following the question, Berm. So you're talking about with inflammatory diseases,
yes,
not hookworm vaccine.
Yeah, talking about the allergic disease like that.
Yep.
And like metabolic disease, like in the developing country, people would have normal infection.
Yes.
So people in that area would have low disease than in the
yes.
Okay.
Yes, those studies really underpinned what we did. So Maria Yazdanbaksh, the group from Leiden University, they've done a lot of work in that area. And it was their early observational studies in areas where they looked at people who had migrated from worm endemic countries to non-worm countries, as well as people in endemic areas who had been treated with anthelmintics. And they showed that their insulin resistance changes. It gets worse when you get rid of their worms. Their allergies increase. There's been lots of studies done with skin prick reactivity to allergens. All of that earlier work predates the clinical trials. Now the clinical trials are really just trying to prove what the earlier observational studies have already shown us.
So if the worm therapy is just suitable for the area that people have low immune to the Helminth compared to the country that have immune response to the worm already. For example, in Thailand, who would be suitable for receive this kind of treatment?
Well, most people in Thailand wouldn't have been infected with hookworms, right? I mean, people in the rural areas maybe, but anyone who has an inflammatory disease and has not been exposed to helminths could potentially benefit. Does that answer your question?
Well, there is one question in the chat box, Alex.
I am looking.
You can read it.
You talked about the rise of Th2 immune response using helminth-based proteins in diabetic patients. Well, it may reduce pro-inflammatory cytokine response to control others, but it is not. But is it conducive and beneficial for people living in underdeveloped and developing nations, where production of type 1 based innate and adaptive immune response is much needed for fighting COPD, TB, and now COVID? That's a very good comment. And interestingly, though, you may have seen a paper. This question is from Alok. Interestingly, Alok, you might have seen a paper recently. I think the journal was eClinical Medicine, which is one of those new Lancet journals, where they showed that people with Helminth infections, people who are naturally infected with Helminths, had a much lower incidence of severe COVID. So you're absolutely right. If you have reduced type 1 immune responses, it may compromise your ability to fight bacterial and viral infections, even though there's evidence in both directions from the published literature that that's the case. And interestingly, maybe you can't fight COVID as well, but people who have worms seem to have less severe COVID, and that's less cytokine storm in the lungs, and probably because of the immunoregulation. I'm not fully up to speed on the TB literature, but I remember a presentation from Tom Nutman from NIH a while ago showing that people who had Helminths did not mount as strong a response to TB vaccination as people who were uninfected. But interestingly, the incidence of clinical TB was no different between the groups. But I haven't been paying attention to that literature, and it may have progressed past then, and it may actually be quite detrimental. I don't know.
Okay, a question, please.
Sure.
Okay, my name is Mubarak, and I'm one of the students from Africa that is studying here in Konkan University. I would like to inform you that we have a group that we carry out a systematic review to assess the level of microbiome research, microbiome gut-brain axis research in Africa. And some of the findings that we have from that systematic review is we notice a trend that gut microbiome communities differ between soil-transmitted Helminth infected and uninfected patients. And what we have noticed is there is a eubiosis, that is promotion of those gut microbiome that are of beneficial effect in the microbiome of those that are infected with soil-transmitted Helminth. So I would like to ask, could you suggest, because we have been thinking about what's the possible mechanism that could lead to that, please could you suggest to us, because we really trying to find out what's the possible mechanism to that, because we are noticing eubiosis, that is promotion of those microbiome with beneficial effect in the gut microbiome of those that are soil-transmitted Helminth patient. Thank you.
Yeah, that's a very difficult question to answer. I think it's multifactorial. So there's not just one answer to that, but there's also this concept of which comes first in this situation, the chicken or the egg. It's clear from animal studies that you do, and experimental human Helminth infection studies, that you do change the microbiome in people once you've given them Helminths. And as you pointed out, in general, the change in Helminth infected people is one towards having good gut species, good gut health, as opposed to poor gut health, like you see in people who have inflammatory bowel disease, for example. Now, it's not clear whether the worm directly induces that change, or the change that the worm induces on the immune system then influences the microbiome to change, or whether it's the other way around. I don't know the answer to that. It's very complicated. I mean, the worms, certainly some of those good gut bacteria secrete all sorts of fatty acids, short-chain fatty acids, and other metabolites that are essential for developing regulatory immune responses. We don't think the worms can generate those short-chain fatty acids on their own. Colleagues have looked to see whether the worms have the genes responsible for producing or for biosynthesizing some of those fatty acids, and at least in hookworms, it's unclear whether they can, even though many authors have reported that these worms can secrete short-chain fatty acids because you can detect them by mass spectrometry. The problem is any of these studies where they look for bacterial metabolites in worm secretory products is you can't rule out the fact that those metabolites came from bacteria that were already in the worm gut. So I'm kind of deviating off topic, but I think it's a very complicated three-way relationship between the microbiome, the worms, and the human or vertebrate host. And I think there's so much crosstalk between all three of them that it's very hard to know. It's very hard to say that there's a single mechanism that results in worm-infected people having a good gut microbiome. I think the evidence is there that it's absolutely true, but as for what's the main driver, I don't have a good answer.
Okay.
I have one more question, Alex. So on the very early slide, you said if you can compare to the non-human parasite, human parasite making as a worm therapy, you said using human parasite would be better.
Yeah.
So if we're going to establish the worm therapy in the animal model in Conchem, and do you think the one that have pathology, but we can optimize to make it low pathology, for example, like Ascaris or hookworm, it would be possible to start with some disease in Thailand? Do you have the idea what kind of disease that we can work on?
What worm would you want to use?
Yeah, I think we can find like Strongyloides stercularis or
Strongyloides is a good one because one of my colleagues here at JCU did an observational study with Indigenous Australian populations who live in quite remote areas in Australia. And some of those populations do suffer from Strongyloides stercularis infection. And there was a clear, and those Indigenous communities also have a very high rate of type 2 diabetes. And she showed that there was a significant protection against developing diabetes in people who had strongyloides. So if you did an animal model, a mouse model of strongyloides, but you'd probably be better using like Strongyloides rati or something like that, right?
Yep.
I don't know if rati is good in rats. I don't know if it survives in mice. Do you know? I don't know. Strongyloides is a good one, but you could also do Ankylostomum solanicum in hamsters, but
we maintain Strongyloides in hamster, I think.
Strongyloides rati or stercoralis?
Rati.
Yeah, okay. But why don't you also do Ankylostomum solanicum in the hamster? Because a lot of these inflammatory diseases, you can induce them in other animal models. So like if you're doing inflammatory bowel disease, there's an easy model, very easy one, which induces acute gut inflammation and then it resolves. And it's just by injecting a chemical into the colon, just via intrarectal administration. That's an easy one to use for developing T-cell mediated gut inflammation. The models we use are for mice, but it would work for hamsters too, I'm sure. And of course, the other good mouse model is either Trichuris murus, mouse whipworm, or Nipostrongylus brasiliensis, which is like a good hookworm model, or even Heligmosomoides polygyrus that Rick Maisels and those guys work on.
Mm-hmm. Any questions? Any more questions? Okay, I think it's 11:20 already. So Alex, I think there is no more question.
No problem. Maybe. Thank you. Thank you for the opportunity to talk to you all, and
I
look forward to visiting soon.
Yep. Okay, Professor Alex will visit Konkan in May. So if somebody would like to talk with him about, to consult about, the research work on this kind of field, you can come. I think you will be welcome, isn't it, Alex? For
yeah, sure.
People.
Yeah, and always happy to have Royal Golden Jubilee scholars too.
Yeah, Alex is happy to have a Thai student as a Royal Golden Jubilee scholarship. He has a student to his lab. I think at least 5.
Yeah.
Yeah. Woodipong is the last student that went to his lab before COVID-19.
Yep. And one of the best ones is on the call here too, Ajahn Suchitra, before she became Ajahn.
Okay, Alex. I'm going to end this session. I would take the opportunity to say thank you on behalf of our department, and we hope that we can invite you to give a lecture as Ajahn again in the near future.
I look forward to it. Thank you.
Okay. Thank you very much.
Bye.
Thank you very much.